Okay, chapter 4 is really long. Okay, so it'll be at least 2 parts to it instead of just one. All right, so okay. Okay, the major learning objective of this chapter really is to understand what a prokaryote is and understand what a eukaryote is and understand the differences between the two. Okay, so prokaryote, on a very superficial level, it comes from Greek words for pre, and then karyote, nucleus. Okay, eukaryote, true nucleus, so it has a nucleus. Okay, all right. So here's the prokaryotes, here's the eukaryotes. Okay. Okay. So a lot of stuff like no membrane, no histones, no organelles. Okay. Eukaryote, membrane, histones, organelles. Okay. Divided by binary fission, which is easier. Mitosis is much more difficult. So in a lot of ways, prokaryote is much more complicated and the prokaryote is much simpler. Okay. Okay, rule number 1 in this class: simpler does not mean better, or more complicated does not mean better. Okay, there are more prokaryotes on Earth than eukaryotes. Okay, prokaryotes do things faster. Okay, more efficiently. Eukaryotes are more complicated, and there's benefits and advantages to each side. Okay, so don't think eukaryotes are better than prokaryotes. Okay. Okay, so the average size of bacteria is 0.2 to 2.0 microns, okay, in diameter, okay, or 2 to 8 micron in length, okay. Okay, most bacteria are monomorphic, so they adhere to a specific shape. Some are pleomorphic, which means they can change shape according to different conditions that they are in. Okay, so some terminology. Bacillus means it's rod shaped. It looks like a pill. Okay, coccus is spherical shaped. It looks like a circle. Spiral. There's vibro. Vibro literally looks like a comma. Spirulum and spirochete are more spiral. You'll see that later. Okay, and then a few are star-shaped or rectangular, but those are more rare. Okay, so this is spirulum, and this is a spirochete. Okay. This is vibro. This is a comma. Okay, this is a star-shaped two star-shaped bacteria. Actually, you can see where they separate in the middle here. Okay, this is a rectangular bacteria. Okay. Pairs is diplo. Okay, diplococci, 2 bacteria that are circles, right? Spheres. Diplobacilli, 2 bacteria that are rods. Okay. Staphylo. Staphylo means a cluster of grapes. So Staphylococci, circular bacteria in a cluster. Okay, Streptococci means chain, so a chain of circles. Okay, tetrads are 4. Okay, Sarcinae, which are more rare, groups of 8. Okay. Okay. So you can see that there. Okay. Okay, so you see that there. Okay, those. Okay. So. Okay. So the scientific name Bacillus should be italicized, right? But if you're just saying the shape, then it should be all lowercase. Okay, so this is anthrax. Okay, so Bacillus anthracis. Okay, so this is also strep. Okay, Streptococcus. Okay, okay. How do you identify Streptococci with a microscope? Make sure you can do that. Okay. Okay. So the structure of a prokaryotic cell. In fact, this part will cover prokaryotic cells. Okay. Okay. The next part will cover eukaryotic cells. Okay. So structure is external to the cell wall. Okay. So Glycocalyx. Okay. Glycocalyx. Glyco, sugar. Okay. Calyx means kind of a vessel. So the external to the cell wall, like a sugar vessel. I don't. Okay, hold on. Sorry. It sometimes just jumps out of my thing. Okay, it's external to the cell wall. Okay, it's what the bacteria is floating around in. Okay, it's viscous and gelatinous. Okay, it's made of polysaccharide or polypeptides. Okay, which is why it's glyco, the sugar. Okay, there are two types. One is a harder one, a harder capsule. It's much more neatly organized and much more firmly attached and plays much bigger role in protection. Okay, it's like a physical structure. Okay, the other type, the slime layer, is unorganized and loose, and it functions like slime. Slimy things are hard to hold on to, so having a slime layer makes that bacteria harder to hold on to or grab onto. Okay. All right, the Glycocalyx can contribute to the virulence. Okay, the capsules prevent phagocytosis. They prevent the cells, the white blood cells, from actually engulfing the bacteria, because they can just slide away. The slime actually allows it to slide a little bit. Okay, and then the extracellular polymeric substance actually helps form the biofilm of multiple groups of bacteria. Okay. Okay, so you can see like the bacteria underneath is a black thing, the bright green thing, the thing coated with GFP is actually the capsule, the slime, the glycocalyx. Okay. Okay, flagella. Okay, so this is flagella. It helps bacteria swim. Okay, so it actually spins, and so you can see hold on one second. Okay, so we'll make sure I share the computer sound. Okay, so
the long whip-like portion of the flagellum is called the filament. Filaments are composed of many identical globular protein molecules called flagellin. Molecules of flagellin form helical chains around a hollow core. Certain pathogenic bacteria can be identified and classified by differences in their flagellar proteins. In contrast with eukaryotic flagella, no membrane covers the filament of a prokaryotic flagellum. The whip-like filament inserts into a curved structure called a hook. The hook is composed of a different protein and connects the filament to the basal body. The basal body is composed of different proteins than the filament and hook. The basal body is composed of a rod and a series of either 2 or 4 rings, which anchor the flagellum to the cell wall and allow the flagellum to rotate 360 degrees. You're viewing a flagellum from a gram-negative cell. A gram-positive cell would only have 2 rings in its basal body, both of which are attached to the plasma membrane.
Okay. Okay. So the different arrangements of bacteria, flagellum, there can be one, okay, there can be on both sides, so they can move through either side. There can be a lot of them, okay, so they can vary in a lot of different ways, okay.
Cells with a single flagellum are monotrichous. When the flagellum rotates counterclockwise, the bacterium runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Amphitrichous bacteria have a flagellum at either end of the cell. Bacteria with a tuft of flagella at one end are called lophotrichous cells. When the flagella rotate counterclockwise, the flagella bunch up and the cell runs. When the flagella rotate clockwise, they separate and the cell tumbles. Paratrichous bacteria have flagella covering the surface of the cell. Runs occur when all of the flagella rotate counterclockwise and become bundled, while tumbles occur when the flagella rotate clockwise and separate.
So that just goes to show you that the bacteria can do 2 things. It can run or it can tumble. So it runs to move in one direction, then it tumbles to change directions. Okay. Okay. So you can either run toward something or away from something. So and that moving away is called taxis. It's the same terminology from which you get taxi. Okay. So negative taxis is going away from something. Positive taxis is going toward something. Okay. Yeah.
Motility is the ability of an organism to move by itself. The motility of a cell may enable it to move toward a desired environment or to flee from a harmful environment. Bacterial cells use structures called flagella, or in the case of spirochetes, flagella-like structures called axial filaments to enable this movement.
So you can see runs and tumbles. Okay.
Although flagella may appear to undulate back and forth like an eel swimming through water, they actually rotate 360 degrees, clockwise or counterclockwise. The basal body spins, much like the drive shaft of an electric motor, producing this motion. When the flagellum rotates counterclockwise, the bacteria runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Movement toward or away from a stimulus is called taxis. The stimulus can be light in the case of phototaxis or a chemical in the case of chemotaxis. In either case, the bacterium contains receptors which send signals to the flagella. In the case of positive taxis, or motion toward the stimulus, tumbles become less frequent when the cell moves toward the stimulus and more frequent when the cell moves away from the stimulus.
Okay. Don't have to know that. Okay. Axial filaments, you do have to know. Okay. Axial filaments are like flagella, but they run along the entire length of the spirochete. Okay. And so, yeah.
Spirochetes are a group of bacteria that are spiral-shaped. They move in a corkscrew-like fashion, using flagella that do not protrude from the cell wall like typical flagella. Lyme disease is caused by a spirochete called Borrelia burgdorferi. Another spirochete, Treptonema pallidum, causes syphilis. Spirochetes have an axial filament that lies between the outer membrane and the cell membrane. The axial filament winds around the outer edge of the cell. The axial filament is made up of a bundle of modified flagella called endoflagella. Rotation of the endoflagella causes the axial filament to rotate around the bacterium, which causes the spirochete to corkscrew through its medium. It is thought that this manner of movement allows the spirochetes to move more easily through viscous human tissues and fluids, such as mucus. Then they can easily attach to the cells below.
Oh, okay. So fimbriae and pili. So fimbriae are like little hair-like appendages, so like that. And that makes it sticky, okay? So they can stick to surfaces better, okay? Pili are something a little bit different. They're involved in motility and not stickiness. There's also something called a conjugation pili, which is involved in DNA transfer, and we'll talk much more about that in another chapter. Okay. All right, cell walls. Okay, cell walls in bacteria are made of peptidoglycan, and they actually prevent osmotic lysis and protects the cell membrane. Okay, so if you ever destroy the cell wall of the bacteria will most likely explode, because usually the bacteria are expanding against their cell walls, and their cell walls actually withstand the osmotic pressure and push back in on the bacteria. Oops. Okay. Oh, okay, got it. So, you can't see me. Okay, that's fine. Actually, I do want you to see me now. So let's try again. Okay, now I can show up. Sorry about that. Okay, so the bacterial cell, okay, itself will expand. Okay, there's more salts inside the bacteria than outside, and usually so fluid wants to run in and expand the bacteria, but the cell wall holds it there. Okay, so if you destroy the cell wall, most of the time what will happen is the bacteria will explode. Okay, so that cell wall helps keep the bacteria rigid. Okay. Okay. So peptidoglycan, know that. Okay. This is what bacterial cell walls look like. Okay. The gram-positive cell walls. You definitely have to know the difference between gram-positive cell wall and gram-negative cell wall. Gram-positive cell wall is a thick layer of peptidoglycan. Okay. Okay. So this is a gram-positive cell wall. Okay. A gram-negative cell wall is much thinner, but it has an outside layer made of lipoproteins. Okay, and lipopolysaccharides, which is actually important because it's involved in the pathogenicity of gram-negative cells. Okay. All right. One of the ways that you can tell a gram-positive cell wall and a gram-negative cell wall is the gram stain. Okay. So crystal violet iodine cells form inside the cell, and we'll do a lot about the gram stain in the lab. So don't worry about too much from the lecture. So just know that gram positive is purple and gram negative is pink-ish. Okay, it's usually pinker than that, but and that's really good. Okay. Okay, gram-positive cell walls. The reason why that's important, gram-positive cell walls are highly susceptible to penicillin, and we'll explain that when we talk about what penicillin is. Gram-negative cell walls are much less susceptible to penicillin. You need a very high concentration of penicillin to make that. Okay, but gram-negative cell walls have something called an endotoxin that gram-positive cell walls do not. That is actually another important thing. Okay, so don't worry about atypical cell walls. Okay, we'll cover that in lab. Okay. So like I said, these guys are susceptible to osmotic lysis without the cell walls. Okay. All right. So the plasma membrane. The plasma membrane is just like ours. Okay. It encloses the cytoplasm. It has peripheral proteins and transmembrane proteins that function across the cell membrane. Okay. So integral proteins, peripheral proteins on either side, and so they control. Okay.
The cytoplasmic membrane of a prokaryotic cell is the main gateway for movement of molecules in and out of the cell. This membrane may also be called the cell membrane or plasma membrane. Cytoplasmic membranes in bacteria are comprised of a phospholipid bilayer. Each phospholipid contains a phosphate head located on the outer part of the bilayer, attached to a glycerol and two fatty acid, which face the core of the membrane. The phosphate is negatively charged and hydrophilic, while the tail portion of the phospholipids is hydrophobic. Thus, cytoplasmic membranes are very effective at blocking passage of many molecules, while other molecules are allowed to pass. Cytoplasmic membranes also contain two types of proteins. Integral proteins often span the entire bilayer and act as channels for transport of molecules that normally wouldn't be able to penetrate the bilayer. Peripheral proteins are loosely attached to the membrane on one side or the other. Some of these may be glycoproteins, which are proteins bound to a polysaccharide. These proteins may function as recognition sites, enzymes, or receptors.
Okay.
The cytoplasmic membrane of a prokaryotic
Sorry. Okay. So this is a fluid mosaic model. I'm sure we already discussed this in anatomy and stuff, so hopefully that makes sense. Okay. Okay. So the plasma membrane selective permeability allows for passages of some molecules but not others. The membrane actually contains the enzymes for ATP production. Remember, bacteria do not have their own mitochondria, right, for ATP production. In fact, the mitochondria are bacteria, and the mitochondrial surface membrane is where you do ATP production. So, yeah, so there's enzymes on the plasma membrane for ATP production. Okay.
With their hydrophobic cores and charged surfaces, cell membranes are very efficient at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, charged molecules and large molecules cannot pass directly through cell membranes, and hydrophobic molecules tend to become stuck inside the hydrophobic interior of cell membranes. In order to allow these complex molecules to enter and exit the cell, organisms rely on two forms of membrane transport: passive transport and active transport. Passive transport occurs along an electrochemical gradient. It does not require the expenditure of energy in the form of ATP for transport. Active transport requires the expenditure of ATP in order to transport molecules across the membrane. Active transport and some forms of passive transport require the use of transport proteins, such as the integral protein seen here.
Okay. So don't worry about chromatophores. Okay. So damage to the cell membrane by alcohols, culinary ammonium, which are detergents, that polymixing antibiotics. Okay, all of that. This is why the hand sanitizer stuff works. Okay, that causes the leakage of cell nutrients, and that's what kills the bacteria. Okay. Okay. Passive process does not require ATP. It always goes from high to low, and there's no energy expended. Active processes, things move from low to high. There is energy expenditure. Okay. Passive diffusion or simple diffusion. That's simple diffusion. Goes from high to low. Okay. Okay.
Passive transport includes diffusion, nonspecific and specific facilitated diffusion, and osmosis. All of these processes occur along electrochemical gradients and do not require the expenditure of energy in the form of ATP. Diffusion is the net movement of a chemical down its concentration gradient, that is, from an area of high concentration to an area of lower concentration. This diffusion requires no energy expenditure by the cell, as the concentration gradient drives the diffusion. Small uncharged molecules, such as dissolved oxygen, dissolved carbon dioxide, small alcohols, and small fatty acids, can freely diffuse through the cytoplasmic membrane. Once equilibrium is reached, molecules will still move across the membrane, but there is no net movement in a particular direction.
Okay. Okay, sorry about that. All right. Facilitated diffusion. This is facilitated diffusion. I'm sure the video already explained that. Okay, so.
Simple diffusion does not require special membrane proteins for movement of molecules. However, other types of diffusion do require such proteins for the transport of molecules. Nonspecific facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of nonspecific facilitated diffusion, these permeases allow a wide range of molecules that meet charge or size criteria to pass through their channel. Some facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of a specific facilitated diffusion, these permeases only allow specific types of molecules to pass through their channel. The channel contains a recognition element. If the molecule is not recognized, the channel will not open to allow the molecule to pass into the cell. Osmosis is a special kind of passive diffusion that involves the movement of water molecules across a semi-permeable membrane. Water passes through the phospholipid bilayer with some difficulty, but transmembrane channels can ease the passage of water through the membrane in a process called facilitated diffusion. Water plays a critical role in maintaining cell integrity. Water tends to move across the membrane from areas of high concentration to areas of low concentration, just like solutes. The difference is that water makes up most of the liquid, so a change in the total concentration of solutes is really the force that causes water to move. If the concentration of solutes outside the cell is high compared to the concentration inside the cell, water may leave the cell, causing the cell to shrivel up. Similarly, if the concentration of solutes inside the cell is high relative to the outside, water may move into the cell and can cause it to swell up and possibly burst.
Okay. Okay, so porins are the channels that allow water to go through. Okay. Osmotic pressure is the pressure needed to stop the movement of water across the membrane. Remember I told you that thing about the cell wall holding the bacteria in? The cell wall exerts pressure which goes against the osmotic pressure. Okay? and that's how the bacteria stays there. The cell wall pushes in, the osmotic pressure pushes out, and they'll cancel each other out, and that's the shape of the bacteria. They destroy the wall, and the bacteria explodes. Okay. So that's osmosis right there. Okay. If you imagine the cell, the thing not changing shape, it is the glass or whatever this thing is that's preventing, that's fighting the osmotic pressure. Okay. Okay. Isotonic, hypotonic, hypertonic. Iso, same as, so concentration on it, both sides are equal. Hypotonic, the salt concentration is lower outside the cell, so water moves into the cell. So if you take a bacteria and put it in fresh water, the bacteria can potentially explode. Sometimes it doesn't because of the strength of the cell wall. Okay, but if you remove the cell wall, it will definitely explode. Hypertonic, this is more complicated. Salts are higher on the outside and water rushes out of the cell. This will kill the bacteria no matter if there's a cell wall or not. So the cell wall holds everything in, it doesn't prevent water from leaking out. Okay. Okay. So isotonic, this is fine. Hypotonic, it could potentially burst. Okay. Hypertonic, it will actually rip the cell, like the cell will shrink and it will rip the membrane from the wall, and it can actually cause plasmolysis. Okay. Okay, active transport,
Active transport requires the cell to expend energy, usually in the form of ATP, to transport materials across the cytoplasmic membrane. This transport occurs through transport proteins and is usually against the electrochemical gradient, like carrying a load uphill. There are three types of transport protein. Uniport proteins transport only one type of molecule. Antiport proteins transport two types of molecules, but in opposite directions. Symport proteins transport two different molecules at the same time, but is coupled with a uniport channel.
Okay. One second. Okay, I think this has gone on for a long time, so we'll stop here and we'll pick this up in part 2. Okay, so.